All right, I think I'm ready. So we got through, yeah, 3 of the 4 categories of macromolecules, right? So we've done carbohydrates, lipids, proteins, and now nucleic acids. We're going to talk a lot more about nucleic acids when we get into actually like DNA and DNA replication, which is further down the line. But this will just be kind of a brief intro into nucleic acids. All right. So what the heck are nucleic acids? Well, their monomer, first off, is a nucleotide. So a bunch of nucleotides strung together makes a nucleic acid. For our purposes, the only polymers are DNA and RNA. These are both nucleic acids. So when you have a bunch of nucleotides strung together, kind of like this, you get a molecule of DNA or RNA. And I'll talk about the differences between those on the very last slide here. Just like with all of the other macromolecules, I want you to know the bond name between the monomers. So each time a nucleotide is attached to another nucleotide, it's called a phosphodiester bond. I'm not going to talk about the meaning behind that, but it obviously involves a phosphate. We'll look at a nucleotide in just a second, and you'll notice that it does have a phosphate group on it. This happens just like all the other polymers that are built through dehydration synthesis. So that's the commonality among all 4 of our macromolecule categories. The polymers are made through the reaction called dehydration synthesis. Definitely review that if you don't remember what that is. And they're broken down through hydrolysis. No matter what kind of macromolecule it is, those are the 2 reactions that make and break polymers.
Dehydration synthesis is when you're adding the water molecule.
No, you're taking the water out.
Oh.
Yep. Dehydration is where you're removing water. Those are really easy to get mixed up. Okay, so let's look at this monomer, the nucleotide, in a little more detail. This is probably, I would say, maybe the most complicated of the monomers for all these different macromolecule categories, because there's 3 distinct parts to it. So we'll start with the nitrogenous base, which we haven't seen anything like this yet. It's basically just a carbon ring. So again, the intersection points here are all just carbons, right? So it's a carbon ring, carbon nitrogen ring, sorry, that's why it's a nitrogenous base. And you can have 1 or you can have 2 attached to each other. I'm not going to get into the difference between those 2, but I'll show you an example where there's a couple, basically a 2-ring nitrogenous base or just a single ring. You'll learn about those when we get into the DNA chapter. And then you have a sugar, some kind of monosaccharide. So we have another one of the macromolecules actually in here. So a monosaccharide, that's a carbohydrate. This just means it's a 5-carbon sugar. It can be a different number. It can be a 6-carbon sugar too. So you don't need to worry about pentose. So we have sugar. And then here is our phosphate group. There's going to be 1 or maybe more than 1 phosphate groups coming off of that sugar. So those are the 3 parts of a nucleotide. So just 1 monomer, which eventually a bunch of these are strung together to make DNA, has these 3 parts. Don't worry about this 5'C, 1'C. I'm not going to get into that right now. So you can ignore those numbers. I just want you to know nitrogenous base, sugar, and 1 or more phosphate groups. That makes 1 nucleotide. All right. We have one nucleotide that's really, really important. This is the only specific nucleotide I'm going to talk about. Otherwise, they're just nucleotides that make up the DNA. This one stands out on its own, and it's called ATP. It stands for adenosine triphosphate. The adenosine part is the nitrogenous base plus the sugar. The triphosphate means there's 3 phosphate groups. So I said there could be 1 or more. In ATP, there's 3 phosphate groups. So the nitrogenous base specifically is adenine. I'm not going to have you guys, there's basically 5 different nitrogenous bases that you'll learn again eventually. Don't worry about that for now, but just so you know where this term, the name comes from, I thought it might be helpful. This is adenine, one of the 5 nitrogenous base options. Ribose, that's our sugar. Remember, that's the 5-carbon sugar that you guys should be familiar with. The monosaccharide, 5-carbon monosaccharide. And then the 3 phosphates, so triphosphate. This is the really important part of ATP, the 3 phosphate groups. And that's because that's what provides the cell essentially with the vast majority of its energy. So ATP is the energy source for cells. You want to know that. And we'll talk about ATP a lot more as we get into the next unit. For now, know what it stands for, adenosine triphosphate. It's not adenine triphosphate, which is really annoying, but adenosine refers to the adenine and ribose. So adenosine triphosphate. And I don't know if I have another slide on this. No, okay. So we'll talk about this being kind of the energy. This is where all the energy comes from in ATP. The bonds between the phosphate groups, when you break those, it releases a ton of energy for the cell. That's why the 3 phosphate groups are really important. Okay, so for now, that's all you need to know about ATP. It's the cell's main energy source. Know what ATP stands for. That's basically it. All right. So that was just one nucleotide, right? That's kind of a weird situation where that monomer, that one nucleotide, is really important on its own. Let's jump into the polymer version of these nucleic acids, which is DNA and RNA. So when you have a polymer, you get the polymer name is the nucleic acid. And here you can see one nucleotide blown up from this chain. But you can see individual nucleotides strung together, right? So they have those 3 parts, phosphate, sugar, nitrogenous base. Those all get strung together. So for you guys on Zoom, this will be a phosphate, sugar, nitrogenous base. And you have groups of those strung together to make the long nucleic acid. And here's an example of a nitrogenous base that has 2 rings attached to it, or made up of 2 rings instead of just 1. Again, don't worry about 3', 5'. You guys will learn that in time. We're not worried about that right now. But it's a pretty complicated molecule because these nucleotides are pretty complicated. And then this is, again, you're gonna learn a lot about DNA and RNA further into the semester. But this is what I want you to know about the difference between DNA and RNA for now, for this exam, just kind of getting into it. So yeah, there's only 2 types of nucleic acids, these polymers, that's DNA and RNA. DNA stands for deoxyribonucleic acid. RNA is just ribonucleic acid. So very similar. The DNA just is missing an oxygen, basically. That's why it's deoxy. DNA is double-stranded, so you've probably seen an image similar to this when DNA is sometimes being referred to. It's a double helix, so there are two strands that are wound around each other into a helix shape. RNA is just one of those. So single-stranded, RNA is single-stranded. And they're obviously very closely related. So what we call the central dogma in biology is that DNA is used to make RNA, and RNA is used to make proteins. So the goal of DNA, all the DNA in your cells, is to eventually code for some protein. Then those proteins will work together to make cells, which will make tissues, etc. But there's a code within DNA. So these nitrogenous bases are basically the code. That will make a strand of RNA. That RNA code will then be read to make a protein. So that's what I mean by DNA, arrow to RNA, arrow to protein. DNA makes RNA, RNA makes proteins. And we will look at that sequence towards the end of the semester and how that actually works. But DNA and RNA are really similar, just a few differences, but they do have pretty distinct roles. Ultimately, the goal is to make proteins. And for now, that's all you need to know about nucleic acids. I know we spent a lot more time on the other macromolecules. That's because we're going to come back to nucleic acids when we get into DNA replication and protein synthesis much further down the line. Okay, any questions about that? That's the end of the material for the exam. So just a little bit of new material. I know it's one class before the exam, but I try not to take up the entire last class before an exam with new material. No questions? Anyone on Zoom? I'm listening to you guys too, I promise. Okay. So, oh yeah. I think this is the same slide as I had on the end of the previous lecture. So just a reminder, this is the type of stuff you should be able to do for the macromolecules. So common features, specific terms, identify them based on some kind of general structure analysis, functional groups, anything specific, like specific macromolecules I mentioned, etc. Okay, so that is the end of that slideshow. And now, I just want to talk briefly about the exam and answer any questions you guys have. So let me give you the basics, and then anything I don't answer, you can ask me. Okay, so we will be taking the exam in person. I mentioned that. It'll be in your biology lab, basically, which I think is 221, but Stewart 221. There's two labs connected, so half of you will be in one lab and half of you guys will be in the other, so everyone can be there in person. So everyone will be in person on Monday. Normal time will start at noon. You'll have 50 minutes to complete it. There are 40 questions total, 40. 35 of those are multiple choice. And then there's 5 fill in the blanks, and that section does have a word bank with it. So you don't have to worry about spelling it wrong or anything. What else? You don't need a calculator or anything. Pen or pencil's fine. It'll be like a Scantron, but a pen will work. You'll just have to, I guess pencil's better so you can erase. The study guide is up and available. Does anyone have questions about the study guide? It's in the module section. Okay. All right, what else am I forgetting about the exam? Any questions? Okay. It's pretty straightforward. You've taken a quiz in here already, so you know the types of multiple choice questions I ask. The fill in the blank is basically multiple choice, just with more choices, right? With a word bank. So same idea. No questions? You guys are good? Okay. All right. So I do want to apologize. My husband pointed out that I scheduled a test for the day after the Super Bowl. I'm sorry. I did not mean to do that. That was not on purpose. And now I feel kind of bad about it, but such is life. Anyway.
Does it matter which lab we go to? Or are we gonna find the lab?
Oh, well, sorry, we'll meet outside in the hallway. Good question. So just line up outside in the hallway outside of 221, and then I'll get 12 people basically in each room. And you'll put all of your material up near the front. So you'll just have a pen or pencil, a water bottle if you want some water. That's it. All right. So how we're going to do this, since I have some of you guys on Zoom and I can't run the Kahoot quiz on 2 computers at once, I'm just going to hold you Zoom people up to the screen so you can see the questions. So let me get this up and going. We'll do the quiz. And I'll kind of briefly explain some of the answers as we go through it. So I'm hoping this gives you a good idea as to how ready you are for, I know you haven't probably started studying all that much, but what? Okay, so there's the sign in. If you haven't used Kahoot before, just go to kahoot.it and type in this number. Sorry guys on Zoom, I'm getting you set up here. All right, there we go. So I cannot see who's answering what answer. Obviously it's anonymous, so no, absolutely no pressure. Okay, I don't think we need the sound. All right, we'll go ahead and get started. If you haven't joined yet, the info will be at the bottom of the screen. All right, here we go. So it's 20 questions. We'll see how many, I guess we'll get through them all. We have half an hour. And I think there's 30 seconds for each question, I think. So the monomer of a carbohydrate. All right, awesome. Monosaccharide. That is hopefully almost a gimme at this point, but you want to know the monomers for all of the 4 categories of macromolecules. Oh yeah, because there's a little pressure because there's a scoreboard, but not really. Which property of water allows it to move from the roots of a tree to the crown, or multiple properties? So the crown is the top of the tree. Oh, sorry, Taisha. Have you still not joined? Try joining again. I don't think I can stop at this point. Just try to answer the questions as we go, even if you're not clicking them in. Okay, so yeah, this is a kind of a tougher one. So in order for water to move from the roots all the way up to the crown, it needs cohesion and adhesion. It needs to stick to itself, that's cohesion, and then it needs to adhere to the side of the little vessels within the plant, that's adhesion. So it has to have both of those in order to make its way up the tree. Which characteristic creates polarity? Nice, good job. Electronegativity. So the more electronegative an atom is, the tighter it holds on to the electrons, right? Making it slightly negatively charged. So you create a polar molecule where you have a negative pole and a positive pole. Great. All right. Which atom in the molecule below has the greater electronegativity? You got a 50-50 shot. Don't let the red and the oxygen, yeah, the red might throw some people off. Should rethink that. Okay, so split. So oxygen is the more electronegative. It's pulling the electrons closer to it. It has a higher electronegativity, making it slightly negative. So you want to know within water, you definitely want to know that water or the oxygen is more electronegative than the hydrogen atoms. So it's going to be slightly negative. And then the hydrogens will be a little bit positive. Number 5. Which of the 4 types of macromolecules is pictured below? All right, lipid, okay. A lot of people got it right, so now let's look at it again. So it's a lipid because it has these really long chains of hydrocarbons. It's attached to a glycerol backbone. So when you see those long chains of hydrocarbons, a glycerol backbone, that is a lipid. This is, I think I took this other question out. It might be the next question. This is a triglyceride. So there are 3 hydrocarbon chains attached to a glycerol backbone. Hopefully that's not the next question. It's the next question. So hopefully everyone gets this right. Yeah, Taisha, it'll be on Canvas later. Nice, triglyceride. Oh, how did 3 people not know that? I just said it. So that's a triglyceride. 3 chains of hydrocarbons, 3 fatty acids, basically, coming off the glycerol backbone. That is a triglyceride. All right. What is the monomer shown below? All right, that's a pretty tough one. So good job to those 7 of you who got it. So it's an amino acid. The nitrogen hopefully indicated to you that that's an amino group right there. So it's an amino acid. It makes up proteins. So it's the monomer of a protein, but I asked for a monomer name specifically, not what is it a monomer of, but the name of the monomer. So that's an amino acid. And then the functional group that is circled. So same molecule. Nice, good job. Carboxyl. So carboxyl, it's easy to confuse with carbonyl. They both have the C double bond O, but the OH makes it the carboxyl group. So the O double bond O plus OH attached to a carbon is going to be carboxyl. You want to know those 7 functional groups, be able to identify them that I gave you in class. All right. What type of bond links 2 amino acids together? Nice, awesome. So peptide bond, very good. Peptide bonds link amino acids together, and that makes a protein. So peptide, protein, I don't know if that's helpful for you, the 2 Ps, but yes, good job. You guys are doing really well. Which of the following is not a characteristic of all living organisms? Nice. You guys all got that one. Awesome. I don't even have to explain it. Not all organisms are independent. Good job. Two or more tissues working together form a what? Nice. Yep. So that's that hierarchy of organization that you want to be familiar with. Multiple tissues coming together make an organ. Molecule below is attracted to other molecules that are what? This requires you to know what the molecule is and some characteristics about it. Okay, yep, so polar is the correct answer. This is water. You want to know what water looks like, and you want to know that it is polar. So it's a polar molecule. Likes mix with likes. So polar molecules mix with polar molecules. Non-polars mix with non-polars. They don't intermix. All right, and yeah, fatty acids are non-polar because they're a lipid. Hydrophobic means it's water-fearing, so it's not going to mix with water. And non-polar, it's not going to mix with a non-polar substance if it's polar. All right. What is this reaction called? A plus B forms a molecule of AB plus water. All right, that's a tough one. So hydrolysis is tempting because you see the water. In hydrolysis, water is a reactant. So water will break apart the polymer here. So if you're taking water out and it's a product, so A plus B, and then you have water as a byproduct at the end, that's dehydration synthesis. You definitely want to keep those two straight. They're very easy to confuse. So hydrolysis and dehydration synthesis, you definitely want to review and make sure you understand what the reactants are and what the products are in each case. All right, so synthesis, that might be helpful. Synthesizing, you're making a bigger molecule. Hydrolysis, lysis means to split. So you're splitting a molecule. All right. What's the monomer of a nucleic acid? We just did this, so may or may not have stuck in your mind yet. Nice, good job. You guys are listening. A nucleotide. Nucleotide is the monomer of a nucleic acid. All right. Which of the following is not part of a nucleotide? Nice, dipeptide, yep. So phosphate group, nitrogenous base, and sugar, those three make up a nucleotide. What kind of biomolecule is an antibody? Nice, yep, protein. So antibodies in our immune system are proteins. It's one of the examples I gave. All right, Brandon's still in the lead. 3 left, 4 left. ATP is a what? What were the other people answering? Nucleic acid, yeah. So those two answers, the blue and the green, are actually, I put that in there to confuse you guys. So ATP is a nucleotide. It is not a polymer, which means it's not a nucleic acid. So it's just a little monomer, a nucleotide, all on its own. Jeremy took over. All right. An atom is more likely to react with another atom when nice. Yes, it has an incomplete valence shell. So the valence shell is the outermost energy shell where electrons are in an atom. If it is incomplete, that atom wants to complete it. So it wants to react with something else where it can take or share an electron. Which of these is a cation? All right, Mg+2 is a cation. So when you see ion, it's something that's charged. So you want to see a plus or minus by the element. Cation means it's positively charged. Anion means it's negatively charged. Let's see, NaCl, it's an ionic bond that creates NaCl. But NaCl itself is not an ion. So if the Na and the Cl are separated, they become ions. But when it's bonded, it's a molecule and not an ion anymore. All right. Last one. Which type of lipid makes up cell membranes? Phospholipids. Phospholipids are the main type of lipid that makes up cell membranes. So I talked about those as an example of one of our lipid types. All right, let's see who won. I guess we came in third first. Allison, nice. Yay. Number 2, Brandon, awesome. Good job, Brandon. Number 1, Jeremy, good job. I don't have any prizes. I always forget to bring prizes for this. I will at some point, I promise. All right. Good job, guys. You guys generally did really well. So hopefully that gives you a sense of what you need to focus on. The majority of the questions will be from, is it chapters 2 and 3, like the chemistry and then the biomolecules. Those first couple lectures are like the nature of science and what properties of life, I think. There will be probably 4 or 5 from that, but the rest will come from chemistry, water, that whole chapter, and then the biological macromolecules. So don't ignore the first part, but definitely know that we spent most of our time in class on those last 2 items. So that's what a little bit more of the test will be coming from. All right, any last minute questions? Otherwise, I'll see you guys. Oh, it's okay. Yep, the chemistry and water, and then biomolecules, yeah. All right, I'll get this link posted to Canvas so you guys can look back at the quiz. And the quiz you took a week and a half ago should be available for you to see. If you can't see it, let me know. And study hard, and I'll see you guys Monday at noon.